So I decided to talk about Nirenberg and Mathai and Nirenberg and Leader's experiments and findings. For a little background, Brenner and Crick first established that the codon consists of 3 nucleotides, and that's them in the picture. You may ask, why is this important? Well, these nucleotides were non-overlapping to ensure that the protein sequences were not restricted. To first start, Nirenberg and Mathai were the first to determine that specific RNA sequence coded for specific amino acids. So when Nirenberg and Mathai began to work together, they had to figure out what the goal or question was. This goal was to figure out the template for protein synthesis, and also could RNA mediate the synthesis of proteins. They began with making 20 test tubes with 19 non-radioactive amino acids and 1 radioactive amino acid to make 20 different solutions of amino acids to test synthetic RNA. One day, Mathai added synthetic RNA, or poly-U, made of only uracil units to the test tubes. This led to activity in the test tube that had phenylalanine. This shows that a chain of uracil gave the phenylalanine instructions and directed the assembly of amino acids into the complex proteins. This was used again in order to identify other codons. Berg and Leader began to work together after the poly-U experiment. They began by using artificial RNA. They made a mixture of trinucleotide ribosome and 20 charged tRNAs, including 1 radioactive amino acid. They put this mixture through a filter after binding was able to occur and filtered out unbound amino acids. If the filter paper was radioactive, this showed that the trinucleotide codes for the radioactive amino acid. This led to the identification of 61 codons and the genetic code that we know today.